Alright, today I'm here with Terry Granshaw from BPS and Tahlia Kinrade. These guys are just going to talk us through the process of what we're doing here today and talk a little bit about what this trial is all about.
So Tahlia, what have we got happening here? We've obviously got a trial and we're biomassing legumes today. Can you explain a little bit about what the trial is and the reason behind why we're biomassing and the process behind it?
Yeah, sure thing. So this is a green waste trial. We're trialling if green waste, which is from the local council. It's made of garden clippings and trees that have been mulched down, has any value as an agricultural input. So we're really lucky here to be collaborating with Sugar Research Australia's Soil Health Project. So we're looking at the soil health implications at this particular site as well. So we've got 3 different treatments and they're replicated across the block. We're trialling green waste, which has just been spread on top and then mixed into the first sort of 10 centimetres of soil. We've got a mill mud treatment, a treatment that's a control with no ameliorant at all, and also another one where we did something kind of exciting, which was mix mill mud and the green waste together. So yeah, that's what we're trialling at this spot.
So we've got soil sample points here that SRA have done previously, and so normally you'd randomly pick spots across the paddock, but we're choosing these positions because we're going to basically lay one over the top of the other to see if there's any differences.
Yeah.
Can you explain the process of how we go about biomassing legumes?
Yeah, definitely. So we're biomassing at the same spots that the soil tests are being done. So we go in and they're replicated across, and we're taking a 1 by 1 metre quadrant of soy plants. We cut them all off just at the ground and count how many stalks there are within that 1 by 1 metre plot as well. We take them back, we weigh the whole amount, then we mulch them up. Terry's on the mulcher doing that.
So we get a wet weight.
Yeah, a wet weight. And then we take a subsample of the wet weight and we weigh that as well. And then the idea is that it goes back to the SRA station at Brandon. It gets dried down and then ground, and then we weigh that again, and then it gets sent off to a laboratory for full analysis.
Alright, so Tahlia, now that the samples have been sent off to the lab, what's the next step after that?
Yeah, so the results from the lab will give us the percentage of nitrogen and carbon that's in the plant tissue that we sampled. And then because we've done the biomassing exercise, we have a good idea of the tons of standing crop that's across the block. And so then we can use those results from the lab to estimate the amount of nitrogen, kilos per hectare, that the soys could be contributing. Of course, at this site as well, the soys are getting harvested, so we'll be taking into account the tons of grain that are getting exported from the block as well.